This meeting is being recorded. Okay. All right, so Beth is helping me out because my camera/computer is not really working, and even when I share my screen, it causes a lot of really weird, glitchy looking colors. Anyone who's here who wants to participate in the pre-meeting chit-chat on iNet totally can. Yeah, just unmute yourself in chat or put something in the written chat. Beth, do you want to like navigate to our project, like the Wisconsin Mycological Society 2026 project? You could look under community if you'd already joined it. Otherwise, we could live demonstrate. Okay, you're in the 2025 project. So if you click all WMS projects that you already joined, this one has all of them. So you can scroll on down and find the 2026 one, but it's gonna be that you have to view more because we're pretty still new in the year. And there it is with 814 observations. All right. Oh, look, right off the top, we've got a Gyromitra esculenta observation. So this is among those we'll be discussing. Do we agree? And if so, why not?
Alden, wouldn't you say that we have Venenata in this region versus Esculenta?
Wow, I
Yeah,
think we have
definitely this is foreshadowing my talk to a degree, but Esculenta is a European species.
Dun,
Right.
dun, dun. All right. So with this, we can demonstrate what to do when you see somebody wrong on iNaturalist. You write them a really mean email. No, scroll down and just is Venenata in Gyromitra?
Yes.
Proper.
I mentioned
Okay.
Venenata.
Venenata. There we go. So suggest an ID, and we could put it down as Gyromitra venenata. Yeah. And then tell you why. You could just say Esculenta is a European name. Now. Or sorry, yeah. Today, Esculenta has become a European name. I don't know. Sometimes I like to write it that way so I don't feel like I'm gaslighting people. Like, yes, your book did say this, but it's different now. Nice. And this would be for anyone wanting to rack up their ID points on iNat. Not that they are redeemable for anything, but if you were to, I suppose, go into Gyromitra esculenta and go through all the ones from North America, you could potentially flip them. And unless they've, I know iNaturalist curators can do that. So a curator would be somebody who has additional power on iNaturalist. But yeah, you can see that Esculenta still has a lot of observations in North America. So you could just sit here and pick them off, I guess, as long as you agreed that they really were Gyromitra venenata and not a different, like Gyromitra or Gyromitra species. Cool. So we got other stuff in the project. People have been seeing Galerinas, Mycenas, Mystery Pezizales from Brennan. What's that? It's flat. It's brown.
He's sitting on a glass slide on my desk, hopefully getting some spores off of it. But,
nice.
It's probably one of those very common ones. I just am never so confident at it.
It's very gelatinous looking to me compared to some that I see.
Well, we'll see. Yeah, but all my stuff's from Ohio now, so it's kind of less. Let's skip over that.
Right. Ooh. Okay. Well, we'll await the microscopy to be added. So heading on back, perhaps. I actually do not recognize Wrinkled Satharella typhracea. Is that
I don't
another
know
thing that we
if
get in
that's-
Ohio and not?
We have it here, Mariah. It's quite common.
All right. Quite common, but that's the joy of mycology. I'm always running into one that I just not familiar. I could see by its common name that at some point people thought it was Satharella, but it is not. Wait. That last bit, Gossipina. I maybe do know this one, but I'm not recognizing the genus. I don't know if that's...
I've got a few observations of it. I actually sent some in on that first plate that Owen has.
Right, the one where WMS is getting some of her specimens sequenced, which is exciting.
Yeah, it looks like there's a total of maybe 5 observations on INAT in Wisconsin of that species. So if it's common, people are not finding it.
Finding
I
it
think
or
it gets
no.
misidentified.
Probably. I mean, what is it? Let's see its characteristics if we can. If we could click in on the genus there, or not the genus, but the species epithet, and read about it a bit more. Yeah, so it is in the Satharalaceae currently. Typhracea. Okay.
I guess I only have one observation on iNaturalist. I think I have a few on MO.
Okay. Checking out the map, maybe we could see where we are in its range. So, like a lot of things, this one does seem to be much more common.
Probably that dot that's right on my house is mine.
Probably. Yeah, zoom in on that bed. Where does Davis live?
Right there.
Right there. And yeah, this one does seem like it has more of an Appalachian distribution with, us being in its range, but
yeah, that's me.
well, right there. So if I were trying to identify this in the field, Tavis, given that you have done so, I see that it is very wrinkled. Does it have the Satharella snap to the stipe?
The stem is a lot more fibrous, and I've got a few photos of it, maybe 10 or 15 different photos of it, and it often has that zonate hygrophony to it.
So hygrophony, as the water is going into or leaving that mushroom's tissue, it's leaving those water spots, those differences in color. And in this case, zonate, it's a bullseye, you could call it. Yeah. And then really wrinkly, big vein looking things. And yeah, that stem does look fibrous, dark.
Yeah, it doesn't do the classic snapperella. Definitely more fibrous.
That's how we knew it wasn't in that genus. Just kidding. Nice moss photography as always.
You got to bring the moss with.
Gotta bring the moss. So given that it looks like you put it as Agricybie to start with, that might be the genus that a lot of people are putting their observations in at first. Because I guess I can see that dark spored, more fibrous stipe.
Yeah, it doesn't feel like a Satharella at all.
Mm-hmm.
In fact, I think my other ones that I have on MO, I may have listed as cyclosomy or agrosemy as well. I don't recall. I don't. You don't need to bring up my MO accounts. I think I've got 5.
Okay. International Man of Mystery in Mushroom Observer World. There. Cool. Well, let's see.
So I'm going to talk about 2 different products. First, we're going to start with our brand new Tesoro. This reel is a full.
Wait a minute. Okay. Wait, Jen, were you saying that's mine? Oh, yeah, you are. Your Flath on there. No worries. That I didn't know that Esculenta wasn't European. I was waiting for the talk to be enlightened about all this stuff. In fact, just today I was telling people about the difference in alleged edibility between Esculenta, which is for sure toxic, and I guess Venenata too, and without a lot of boiling and stuff I don't even want to mess with, and some of the other species. I don't know if, Alden, if questions about edibility are fair game in your talk, or like in the Q&A. If you wanted to rule them out, that's cool too, because I know there's like disagreement, but.
Yeah, no, they're absolutely fine. I feel like I can speak with nuance that gets to the truth, which is sometimes vague.
Yes. AKA, you got a PhD, you're a scientist. Very cool. Okay. Neophavulus, beautiful. And I think this is something where I'm going to maybe test my ID knowledge. I had been informed by somebody else on iNaturalist that the way to tell the difference between Neophavulus americanus and Neophavulus alveolaris, which is apparently still in North America, but we just split out Americanus, is that the Americanus one is paler on the top without sun bleaching. This is, of course, a naturalist-based hearsay, and not me actually going back and reading the description. But
Yeah, actually, with Neophalus americanus, that's going to be a species group containing several species.
okay, and you think
Not listed
alveolaris
as such as on INAT, but it does require some more delineation.
okay, so maybe it's really TBD, and you could, and whether you put alveolaris on it or Americanus, you're not.
You can put Americanus in that, I suppose, because it'll be in the Americanus group, although alveolaris probably does exist here. I think it has been sequenced here. But for the most part, what you're seeing is
Yeah,
a complex of species.
yeah. So maybe just put it up as Americanus, and for
I
this one, it's
would,
pale. So we would do that. If they want to change it to a species complex later, by they I mean like the iNaturalist curators, they can do it.
you can add in the comments to Neophavulus americanus group just as a comment.
Yeah.
That would certainly be more accurate. It's not going to be a singular species, that's for sure.
Hmm.
No, americanus group.
I like Neophavicus. That definitely sounds like a genre that would get made up.
I believe somebody was working on this at least maybe a couple of springs ago, a couple of years ago. Somebody was trying to do some division there, and I don't know that they're going to be readily separable on morph. I think it's going to
Yeah.
require a little bit beyond that.
Yeah.
So Neophavulus americanus group is going to be as close as you're going to get in the field, as far as I can tell.
You want to look at that Rushla Brennan found so we can maybe make Tavis idiot or, comment.
You can take a peek at it.
Yeah. It's nice and
Yeah. Did you
pale.
find that this spring, Brennan?
Found it today.
Found it today. So there's only a couple that grow in the spring. Vinacea would be one. It's awfully pale for that. Did you look at the gills?
Not closely. I didn't take it, but I think the next photo shows the underside. Right, Beth?
Yeah.
Oh, I
No.
have two observations. Maybe I don't have the underside on.
One of these that are potentially in subgenus Heterophilidae, you would want to check for gill forking.
Okay.
And where those gill forks are, if they're at the margin, if they're at the stem, or if they're in the field.
I do have a second observation of probably the same species. If you want to look at the gills, it's on a different one, so you'd have to go back.
Yeah, it looks pale for Vinacea, but you can't judge a Rushla by its color.
Oh man, you can't see
There's
gills
too much color variation within each species. I mean, some that can go from purple to yellow and be the same species. But looking here, I'd say it might be in subgenus Rushla itself. It might be something around that Gracilis fragilis clade.
One of them was almost certainly associating with yellow birch, or possibly a lot less likely a white oak.
Yeah, that really doesn't help in this case.
All right. I remember I'm in Ohio, so there's also
Yeah, yeah.
I'm ahead by like a month.
Oh, that's true too. Well, even then, I mean, even July here is the only russell we're really going to see is Vinacea and a couple of the really small, fragile ones, Nobilis and uh, Miniata and Vinacea, of course. Fragilis to an extent,
I
and
feel like
Fragilis again is a complex.
I was gonna say I feel like I've seen some of the shrimp russula earlier, but I don't actually recall unless I were to go and look at my naturalist again.
This is
Like so,
almost certainly not one of those.
yeah. I was just thinking of other early things, but it doesn't look like that at all.
So, Russula Barley is one of the ones that we would see in the eastern United States, and it's going to be a lot oranger than the purple ones that we see more in like the Rocky Mountains.
Yeah,
So the
it
shrimp
was
Russula I find here in northern Wisconsin are almost always orange, looking very similar to
interesting.
Russula decolorans.
Okay. I don't know if this works for you, Beth, but I'm gonna put one of my observations in the chat and you maybe could open it and that observation. Getting like free help from Tavis Russula expert guy here. And if anyone wants us to look at any particular observation of their own or anybody else's, you could put it in the chat too. So this is one that I ID'd as Russula Zeromphalina.
It's not.
Ding.
No, that's Marie.
Okay. I thought the Marie blush was supposed to be subtle.
It's
That's the
not.
okay.
No. So there's another species I can't quite recall its name now. There's another one that looks very similar, both in the Heterophylidae and both with a prunos bloom on the cap. If you look close at that cap, it should look dusted.
That does look dusted. It looks like a plum. The prunos.
I reached this on the right side of that cap. That's your prunos
Yeah.
bloom. Yeah, that's
Yeah.
absolutely Marie or close. It's definitely in subsection Amininae of the Heterophylidae.
And those would smell shrimpy too.
They do not. They should smell just like um, a pleasant mushroomy odor.
That's interesting
The
because
shrimp
it did
russula
have
has a very almost foul shrimpy odor, especially when dried. They get quite the
well, and see
stank.
that's the thing. And I think that's what was leading me in this direction was that it had such a distinct fishy odor and it was rank and it was strong when fresh and when dry.
You weren't smelling
And
decomposition?
no, I no.
Yeah.
And I I am confident on that because smells is definitely one of the things that I think about a good amount. And I thought about it a lot with this one while I was trying to ID it. As you could see in the last picture there, I actually did take a little tube, little genetic, a little piece of it to do sequencing on. It was before I was doing good labeling, so you could see my really terrible labeling there, where instead of giving it a code, I tried to write shrimp.
All right. Yeah,
And
it's
don't be
definitely
like,
not a shrimp russula.
yeah,
It's Marie or there's another species too, but I think we see that more out east, and the name is escaping me right now.
all right. Well, yeah, feel free to pull the trigger, Beth, and disagree identification. That'll help me remember to go back to it. And figure out. I haven't done the sequencing on that because it's not really been a priority for a minute, but now it is. Now that it's
Yeah, another thing you'll see with all of the shrimp russula is uh, a faint blush on the stem, but discoloration, kind of a grayish when you rub it, especially towards the apex of the stem.
that's interesting.
And Marie is not non-discoloring.
Okay. Yeah, I don't remember it discoloring at all. So that is in line. I do wonder, we've talked about this before with some fungal smells, that they just might not be as well documented as we would like. That, like, what if there's variation within a species even, or like we know that there's variation with some traits, like level of bioluminescence from population to population of things that have bioluminescence. So why not other traits?
That's a good point. A good example would be the Omphallotus that was found at Arnie's last year that glowed brilliantly bright blue rather than the haunting green that we typically see in pictures.
Mm-hmm.
I mean, it looked like blue headlights coming through the woods.
Yeah. What's that kingdom fungi one there? Somebody's just being. Sometimes I do that when I upload things. I just put everything up as kingdom fungi, but this one might also just be an observation of the brown cubicle rot there.
Yeah, that might not even be fungal.
No, I mean, I guess as far as I know, brown cubicle rot would be fungal, but I don't know if you could ever, because it's somebody who can break down.
Yeah, no, that looks fungal, but sometimes you'll see it in red oak where the medullary rays will actually dry to white and look fungal, but it's just cellulose.
Oh, I see. So you could just have something that's even okay. Well,
It's fungal colored, and that's where it ends.
yeah. Good luck to this person in IDing it. It might be a forever kingdom fungi. But I have some like that too, where it really becomes just a record for me of what I saw, even though I can't really say what it is. Let's see if there's anything else. We're at we have about 4 minutes before Alden's gonna talk. Oh, here, go ahead and last one. Click on the Schizophilum commune. It's just a beautiful photo. And I think this is my mom's picture, so also happy Mother's Day to my mom.
Oh, very nice.
She found these and photographed them, and this just goes to show that there are extremely common fungi that are just so photogenic and beautiful. That's what it looked like from the top when she picked it up, so not too much to write home about. But then you flip it over and you can get these some of the larger fruitings of Schizophilum, or how do my friends say it? I can't say it that way. I say Schizophilum. But yeah, gorgeous. Looks like a sea creature to me, or like a seashell. And that was seen at our Dr. Tom Volk Memorial Foray in Perot.
You have a species list from that, Mariah?
I do, and thank you for reminding me to send it. I will after this. Do have a species list. So we could go ahead and quit. Thank you so much, Beth, for taking over and doing the clicking through bit for me.
Anytime.
Uh-huh.
Would you like me to stop sharing?
Yeah, and then that can also give Alden a chance to maybe try to start sharing and get things set up, because I'm just going to go ahead and read her bio. Introducing Alden. Okay. So
you on my screen, you said?
Yeah, you could get that going because a lot of times in case there's anything going wrong, then we can figure it out. But it's probably going to be pretty quick.
Can you all see that?
We can see it, or at least I can, but it's showing the Zoom. It's not showing like a presentation. So here we go. Now you can figure that out, and I will read your bio. Okay. Dr. Alden Dirks has been a practicing mycologist for over a decade. They are currently a postdoctoral research fellow at the Children's Hospital of Philadelphia, where they investigate antifungal resistance in invasive fungal diseases affecting immunocompromised children. Previously, Alden earned their PhD in ecology and evolutionary biology at the University of Michigan in 2024, and a Master's of Science in agroecology from the University of Wisconsin-Madison in 2019. While fascinated by all aspects of fungi, their primary research interests include fungal systematics, especially pertaining to lorchels, our Gyromitra and friends, crust fungi, and fungal dark matter taxa, so mysteries, fungal mating systems, and queer mycology, fungal genomics and secondary metabolites, and medical mycology. Alden is also an avid forager with a lifelong goal of eating 1,001 fungal species. As an ambassador of the Kingdom Fungi, Alden teaches the public about mycology through forays, workshops, and lectures exposing students to the lesser-known and underappreciated organisms that Whoa, getting a little bit of feedback there. In any case, a lot of interests. I feel like this is what happens when you keep going deeper and deeper into mycology. You're like, I like every part of it, actually. Yeah, so that's a really cool background. I hadn't known you were moving into like medical stuff, but very, very cool. So welcome so much, Alden, and we're super looking forward to hearing from you.
Thank you so much. Yeah, I'm still trying to figure out how to share my screen, so I just asked. I just changed to the app instead of the browser.
Okay. Yeah, I mean, like, screen sharing and just my camera in general failed for me today too, so I don't even, and Tavis as well.
I could see your presentation before when Mariah
Really?
said she couldn't see it. I could see it. He had a picture. Yeah, so I think it was working. It just he
Okay,
couldn't see it.
so maybe it is a problem on my end. You go ahead and just set it back up maybe as you had it, and I'll find a different way to join the meeting.
Can you give me admin access again?
Yes, let me look to make
There's
sure.
not two of me.
You should still be a co-host. Oh, maybe you had, here. You are now once again a co-host. There
Okay.
you go.
I can see that.
Okay, great. Perfect. Yeah, it was just me. And I think I can see it now too. Don't know what was happening to me before. Anyway, thank you. That's a gorgeous photo. And yeah, without further ado.
Thank you. Why is this such a struggle? How do I make it? Okay, there we go. There we go. Okay. Is that now full screen for everyone?
Yes.
Okay, great. Thank you so much. Yeah, and thank you for that introduction. And it's nice to be talking with the Wisconsin Mycological Society again. When I lived in Wisconsin, in Madison, I gave a talk with the Wisconsin Mycological Society back in maybe 2018. So it's been 8 years or so. And I have moved into the medical mycology space, which is strange and exciting for me, working with fungal infections of children, not doing anything clinical, just doing research on the actual fungi and the evolution of antifungal resistance in fungi, just like we have antibiotic resistance in bacteria. We also see that in fungi as well. But for this talk, I'm going to go back to my PhD work, which is on this group of mushrooms that I like to call lorchels. And it's entitled False Morels or True Lorchels, a deep dive into the toxicology and taxonomy of Gyromitra mushrooms and related species. We are at the tail end of the lorchel season in Wisconsin, but depending on where you're at, you can still find them. And actually, I know some people in the society just found some today or yesterday, as we saw. So this is seasonally relevant, and I hope you learn a lot that can help you this season and also going forward about this really interesting group of mushrooms. So to start, when we think about the spring, one of the most iconic fungi are the morels. People get really excited about these. They pay a lot of money. They go hunting for them. And these are considered choice edible mushrooms in field guides. This is often contrasted in field guides with what are called the false morels. These are Gyromitra species, and these are regarded as deadly toxic. In this talk, I'll be referring to these false morels as lorchels. Lorchel is a name that has been applied to these mushrooms by experts in the group, like Nancy Smith Weber, daughter of Alex Smith, for example, who studied morels and Gyromitra mushrooms a lot. And it's been used by other people as well. So this is kind of my rebranding campaign. I want these mushrooms to be not what they're not, but something of their own, so lorchels. However, this simple narrative, this simple binary of good and bad, is actually much more complicated. In fact, we see that morels can actually be deadly. Uh, Infamously in Bozeman, Montana, I believe it was. No, Idaho. Sorry, I'm forgetting right now. There was a really bad poisoning event that happened where morels were served raw at a sushi restaurant, and dozens of people got sick and a few died from eating these raw morels. So it's been known for a really long time if you're eating morels, you have to thoroughly cook them because they do have some unknown toxin that seems to be damaging red blood cells and can actually make people very sick or even kill them. And on the other side, with these lorchels, there's actually a long-standing culture of people consuming lorchel mushrooms. And that's what I'm going to show you here in this video, which is a nice little food video from Finland, where you can see the Gyromitra mushrooms being cleaned, boiled for 5 minutes. They get rinsed thoroughly with water, and they get boiled a second time, rinsed once again, and chopped up. And here they're making it into a nice little cream sauce and turning that into an omelet. So I don't know if that makes anyone want to eat these mushrooms. It certainly looks nice to me. Uh, but the reality is, these mushrooms, even though they're eaten as a delicacy in some parts of the world, particularly in Scandinavia, they do have a mycotoxin in them, and it's known to be a very deadly and potent mycotoxin. And this mycotoxin is called gyromitrin. So if one is exposed to gyromitrin, symptom onset occurs about 6 to 12 hours after exposure. And what we call the common and mild symptoms, put mild in quotes because it's not a pleasant experience, is vomiting, abdominal pain, and diarrhea. And in more severe cases, one can experience neurological symptoms, progressing to seizure, hepatic coma due to free radical damage, destruction of the liver, and then death as well. And something that's been emerging over the past few years is the potential for these mushrooms or the mycotoxin gyromitrin to cause cancer and neurodegenerative diseases such as ALS. So here on the right, we have a photo of a community in the French Alps, Montchavin, if that's how you pronounce that. And there was a study done in 2021 where they found that there was a cluster of people with neurodegenerative disease in this alpine community. And they linked that to their consumption of these lorchel mushrooms with gyromitrin. And the thought was that they were not prepared properly, they were exposed to gyromitrin, and this actually caused over time neurodegenerative diseases, a really large cluster of people with neurodegenerative disease in this community. So gyromitrin is not to be trifled with, in summary. What is gyromitrin? So the actual molecule is acetaldehyde N-methyl N-formylhydrazone is the name of the molecule. And this refers to this first substance here. There's what we call 8 higher aldehyde homologs that exist in these mushrooms that you might collectively call gyromitrin. Even though this first substance is primarily what we refer to as gyromitrin, you can see that there are these homologs with varying length side chains. Okay, so chemically, what this means, the gyromitrin is water soluble, it's volatile, and it's acid and heat labile. And that's why you can actually cook the mushrooms because you can cook away the gyromitrin to render the mushrooms safe to eat. When this gyromitrin is exposed to heat and acid, for example, if you're cooking in a hot pot, it breaks down into N-methyl N-formylhydrazine and finally into monomethylhydrazine. So this is the primary toxin. Monomethylhydrazine is actually used as a rocket fuel, and it's been known to be very bad for you, very toxic for decades because of its industrial uses. And this might be the only mycotoxin, at least the only one known currently, that can poison you via inhalation. So even in the preparation of Gyromitra mushrooms for consumption, you have to be careful about breathing in the fumes. So it's pretty intense, honestly, that process. And when I entered my PhD, there was not a lot known about this. There was a lot of speculation about gyrometrin over the decades, but not a lot of solid data and evidence. And that's because the group that the Gyromitra mushrooms exist in, this family Discinaceae, is quite complex. There's a lot of different species, and we didn't know a lot about the taxonomy. There was a lot of we were missing. In particular, there were some old inadequate species descriptions. A lot of these mushrooms were described centuries ago, and it's really not quite clear what species people were referring to over the years. The mushrooms are frequently misidentified in Fungaria and in the literature. There's a lot of unclear species boundaries and inconsistent species concepts. And finally, a lot of unverifiable poisoning reports and chemistry studies. So a lot of noise, but very little strong, clear signals. I'm just going to go back here just to show you this really amazing diversity within this family. We often think of just the Gyromitra mushrooms, but we have these cup fungi that we might also call discoid fungi. We have truffles, shown here, and then we have other stipitate fungi that I will also call lorchels that have this central stipe and this kind of cerebriform or brain-like cap. So we have a lot of morphological diversity in this family. And what I'm going to be showing you here is what we've learned about the taxonomy and evolution of the Discinaceae family using molecular phylogenetic analyses. And typical, or maybe in the past, the primary way in which people did phylogenetics was using a single gene or maybe a few protein coding genes. And you take this genetic information and you're inferring an evolutionary history. So I will be showing some phylogenetic trees. It sounds like y'all as a group talk about them, but just to review real quick what a phylogenetic tree is, here we're seeing what's basically a graph showing the evolutionary relationships between different taxa. Could be different individuals of the same species, different species, populations, whatever. So here we're seeing those taxa at the tips of these branches, and this is referring to the species present in the current time. And these species or taxa, they're connected to each other by branches. And as we go from the right to the left, we're actually going back in time. So here we're seeing that taxon A, taxon B share a more recent common ancestor, which is what's shown here as the branches come together at a node. These are the ancestors of the species compared to taxon C, for example. And we can see if we go all the way back in time, there's a single ancestor that gave rise to all these different taxa. So a brief overview of a phylogenetic tree. Um, Recently, in the past few years, I would say, maybe in 10 years at this point, but it's increasingly so now with the availability of large data sets and genomes, we're moving from single or multi-locus phylogenetics to phylogenomics. So instead of using one or a few genes to infer evolutionary history, instead we're using hundreds to thousands of conserved genes across species. And what you see is sometimes conflicting signals between those genes, and you have to basically average out those signals to try to infer how species evolved over time. It's a much more statistically complex analysis, but instead of just looking at the evolutionary history of a single gene, you're kind of looking across the whole genomic context to infer how species evolved. And that's what I'll be sharing with you today is my work using phylogenomics to better understand the evolutionary history of this family Discinaceae and the things we're able to learn about the evolution of Gyromitrin, as well as certain trophic modes and ecological guilds based on this genomic information. So some of the questions I'm going to address in this presentation include the evolutionary history and taxonomy of the Discinaceae family. We're going to talk about their trophic modes or ecological guilds. In other words, what they're consuming, how they're making their living. Which species produced this mycotoxin Gyromitrin and how did it evolve? How many species are there in this family and where are they found? And finally, how you can participate in the documentation of Discinaceae fungi and really other fungi in general. It's going to be kind of an overview of some of the resources available to community scientists. So before I jump into the phylogenomics, are there any quick questions, or maybe I'll just see if there's any comments?
There is one in the chat that's more about, you might be able to see from Jeremy, that's about sort of the aqueous extractions and the toxins. I don't know if you want to pause and talk about that. Totally good. If you want to
Yeah,
do
I'm
it.
sorry. I'm actually we've been using Zoom for years, but I still struggle so much. Could you
Okay.
yeah, read the question? Oh, here's chat up here.
Yeah, I'll
Um,
read it aloud too for the video's sake. So Jeremy said Volk used to say people claimed it was safe to eat if you did aqueous extracts with gallons of water 3 times to leach the toxins. Do you have any thoughts as to whether the advent of the ALS effects that have started to be documented are due to residual toxins in the fungal fruiting body on oral consumption versus toxicity D/T? I actually don't know that notation. The volatile compound you described before.
Yeah, so the thought there is people have been consuming these mushrooms for a really long time as a delicacy in Scandinavia. And in Scandinavia, you don't see a higher rate of ALS that you would think would be associated with the consumption of these mushrooms. So the analyses that have been done, the quantitative analyses, do show that when you prepare these mushrooms properly, um, some people might argue there's no proper way, but if you prepare them in the traditional way of boiling them thoroughly, tossing the water, boiling them again, and not just like the water is starting to like steam, but it's a rolling boil, it's a high temperature, it's 100 degrees Celsius, that you're getting rid of the vast majority of the gyromitrin. And the people who've been consuming these for a really long time, we don't see the higher rates of ALS in those communities. So the thought is that it really is improper preparation, and it may have been this community in the Alps. There wasn't the same culture for people to know how to prepare them properly, or it was a higher altitude elevation, and water boils at a lower temperature, which is another consideration. If you're not boiling at 100 degrees Celsius, but 98 degrees Celsius, you might get rid of less of the toxin. So. Yeah, long answer short. It seems with proper preparation, people are safe eating these, but it is risky, and you do have to know what you're doing. So I've certainly never consumed them, and I wouldn't quite recommend them, but to each their own. Um,
All
yeah,
right, cool. Thank you for pausing and answering that. I'm looking
yeah,
forward to the phylogenomics stuff though.
for sure. So what I'm going to share with you now is work that was published in 2025 in the journal Molecular Phylogenetics and Evolution. And what we did here is I sequenced 62 lorchel genomes, lorchel in the broad sense, really species from Discinaceae, including the cup fungi, the stipitate lorchels, as well as truffles. And we took 1,500 genes and we did multiple different analyses on these genes, and we inferred an evolutionary tree of phylogeny for this family with really strong support. So one of the ways that we measure support for an evolutionary tree is bootstrap. But in phylogenomics, that's typically maxed out because it's a measure of variation. So you use other measurements like gene concordance, which is shown on the branches here. And I know that's a lot, and we might have to spend a lot more time talking about the details here, but in sum, this was a really high quality evolutionary tree. And for the first time, we're able to infer the evolutionary relationships between these different groups in the family. So what I'm showing you here on the top is we have the Discinaceae family, and then we have our outgroups, which include the morel family, Morchelaceae, as well as the main truffle family, Tuberaceae. So as I said, we resolved the early evolutionary history of lorchels. All branches had high bootstrap support, but looking at this other measure of gene concordance, which is basically asking how many of the underlying gene trees showed the same evolutionary topology, we found that it was generally very high, but there were some nodes with still a high degree of uncertainty. So what we're seeing here is that we're basically hitting the limit of what we can infer about the evolution using genomic data. And that's because when you have really rapid evolution, then you might not have a strong signal within the genes to actually infer evolution. So we're kind of hitting the limit of what we can infer. However, this still is the most well-supported tree and in general, highly supported. With this evolutionary tree, we did some revisions on the taxonomy of the Lorchel family. As I'll talk about later in this lecture, we recognize two different tribes called Discinaceae and Gyromitriae. I didn't invent the use of tribes. These were pre-existing categories in this family. And we recognize 10 different genera. So there was a lot of hoopla, I guess you could say, when this study came out. There's a lot of conflict in taxonomy around lumpers and splitters. And I think some people are upset about the splitting of what used to be called Gyromitra in a broad sense into 10 different genera. And we're going to talk about these different genera, and I hope you can find the utility there. But I do want to say that however you feel about this, utilizing taxonomy and nomenclature, there's certain rules of nomenclature that have to be abided by, and as well as taxonomic principles of monophyly. So in some sense, there was not really any good alternative to the taxonomic approach that we took. And if you want to know why, it's pretty much because of this genus Hydnotria, which is truffles, that is closely related to Gyromitra. And there's basically no way to follow the rules of nomenclature and to recognize Gyromitra in a broad sense. And I go in a lot more detail in the paper, but yeah, we're going to talk about these genera and see the utility in them as well. I also want to say that actually these genera are consistent with divergence times for other genera. For example, if we look at time in the past and we think of a genus as evolving 50 million years ago or whatever it may be, these time frames have been applied to different groups, for example, morels. And we see that this taxonomic system is actually consistent with divergence times for other genera, including the genera in the Morchelaceae family. Anyway, so we're going to talk about these different genera after I go through this portion. So from this work, we also discovered that some species previously thought to belong to this family formed a well-supported group constituting a new family. Here I'm showing you Paradicina donadinii and Paradicina melaleuca. So Paradicina melaleuca used to be considered a Discinaceae fungus, but we found that these two fungi and an additional one were in a family sister to the Morchelaceae here. This family doesn't yet have a name, but just so you know, there's actually an unnamed family that we recognize as existing. And we also saw that the morphology of the Discinaceae fungi is all over the place. So if we're looking here at the discoid fungi, stipitate fungi, and the truffles, we see that there's not a clear monophyletic or single evolutionary event leading to these different morphological groups. And perhaps this is not a surprise because we see mushroom macromorphology as a dynamic trait with lots of convergence across kingdom fungi. If we think of guild mushrooms, for example, they've evolved again and again in different orders of the Agaricomycetes class. We have guild mushrooms in the Rucellales, in the Garicales, of course, Bullitales. So we see the same thing in this family. Discoid fungi appear in different clades, as well as stipitate fungi, and yeah, which is consistent
I,
with a lot of different groups.
I have a quick question about this, if that's okay.
Sure, yeah.
I also want to apologize for coughing. I was like unmuting myself to interrupt, but then didn't mean to. So, but my question is about the end of your tree there with the outgroup of the Tuberaceae. I'm just super curious what the discoid Tuberaceae is.
Yeah, that's I believe the Coromyces venosus.
Really? Okay.
Yeah, yeah. So you see this within the Pezizales, and you find stipitate forms, discoid forms, truffles. They just keep evolving again and again in different clades.
I had known that species more as a truffle, but I would not put it past fungi to have, multitudes to their form. So we could come back to that later. But
No,
yeah.
I think you're right. I think you're right, actually. I don't remember. I'll have to find that information. I think I had this in the original publication as just truffles, but then I think it was Greg Benito, who's a co-author in the paper, who informed me that there are discoid species in that group.
I'll have to take it up with Greg. He's on my committee, so he owes me emails.
Yeah, yeah, Greg could definitely tell you. But you're right. Coromyces is, I believe, a truffle, and I'm not sure what the discoid species is. So
We'll find her
I'll have to
later.
look
Okay,
into that.
thank you.
Yeah, so we see this consistent with other Pezizales groups and just fungi in general. Macromorphology, which used to be the main taxonomic signal used to identify and to group mushrooms together taxonomically, is convergently evolved. It evolves independently again and again in different lineages. And we see that here with Discinaceae. So with this really good phylogenetic tree, let's look at Gyromitrin, the mycotoxin, as well as the ecological guilds. And this information comes from two different studies, one from 2020. When did I publish this? 2020, I forget, 2023.
Or
And this other one from 2024, looking at the actual distribution of gyrometrin using these gyrometrin assays to determine which specimens and species produce this mycotoxin. So we're going to take those data and apply those to the phylogenomic tree that I just showed you. And what we're seeing here is that the taxa that are colored in blue are ones in which gyrometrin was not detected. And the ones in red are the taxa in which gyrometrin was detected. So it was no surprise that we detected gyrometrin in the genus Gyromitra, shown here, because that's the classic iconic group that produces gyrometrin. That's the group from which gyrometrin was first isolated and identified. But to our surprise, we didn't actually detect gyrometrin in any other clade besides a discoid clade called Pezizales, these cup fungi here. And this was a surprise because people kind of conjectured and hypothesized that other groups were toxic and contained gyrometrin. And no one, as far as I'm aware, has tried eating these cup fungi, so this was a complete surprise for us. So this distribution was really interesting, and it suggests perhaps two different things. That gyrometrin may be a result of horizontal gene transfer, which means that the genes that are responsible for producing gyrometrin were not acquired through descent over time, but rather they were transferred from distantly related species. And that could have been a horizontal transfer from the genus Gyromitra to Pezizales, or Pezizales to Gyromitra, or from a completely unknown other clade. And we see this happening a lot with secondary metabolites that are ecologically useful. Maybe the prime example is psilocybin that's been horizontally transferred between various guild mushrooms, for example, from psilocybe to paniolus to gymnopolis. So those mushrooms are distantly related, but they share the same genes because they actually got transferred between unrelated groups, which is a fascinating process that we still don't understand well how that actually happens in fungi. Alternatively, this could be a result of convergent evolution. Perhaps the genes that make gyrometrin are conserved across this group, but it only requires a little bit of a tweak to make gyrometrin, and that happened twice in the family. And we think that these explanations is likely because it's less likely for there to be so many loss events, which would be required to explain this distribution if gyrometrin was acquired, was gained in the common ancestor of the family and then acquired by all the descendants. In that case, for it to be absent from all these different clades, there would have to have been a lot of loss events in the evolutionary history, which is just less parsimonious, less likely. But this is still an open question and one that I would love to hopefully answer sometime in the future. And the true story is likely much more complicated. There could have been losses, there could have been horizontal gene transfer, there could be related genes in these other clades, and they're producing some other toxin. So it's a really exciting story that we're hopefully going to answer at some point in the future. And as evolution goes, it's rarely a nice clean line, and I'm sure it's going to wander all over the place. And with these genomes, we're also able to peer inside the genomes to infer something about the ecology of these fungi. It's been an open question for a really long time. What exactly are these fungi doing? People will find these really iconic Gyromitra mushrooms. They'll find the truffles, the discoid fungi, and it hasn't been clear exactly what their ecological role is in the environment. So what we found here, looking at the genomes, is that there's a strong signal of the truffles in the genus Hypnotria being mycorrhizal. And there's this classic signal that you see in genomes for mycorrhizal fungi, where they have enlarged genomes due to the result of a proliferation of these transposable elements, which are selfish genetic elements, as well as a reduced casein content. So let's walk through that. This first part here is showing genome size. As you see, we have a significant enlargement of genome sizes within this clade consisting of the Hypnotria truffles. We also see, and this is driven by transposable elements, which is the size of these dots here. We also see a reduction in cazymes. These are called carbohydrate degrading enzymes. And this makes sense because if you're a truffle, you're relying less on consuming the food around you and more on acquiring sugars from your host, from your plant host. So again, another common signal for mycorrhizal fungi, they have a reduction in these enzymes that are responsible for breaking down carbohydrates. And perhaps surprisingly, we actually saw that there's pretty much a saprotrophic genomic signature to all the other clades. Saprotrophic just meaning they're breaking down dead organic matter in the environment. So again, in summary, Hypnotria had a genomic signature of being mycorrhizal, which we knew already. We already knew that they associated symbiotically with the roots of trees. But all the other clades in the family seem to be saprotrophic.
I have a question from the chat briefly on this graphic, which is so red is detected, like Gyromitra was detected and it's present, and blue is not present. And what is the white or non-colored ones?
White is just ones that were not tested.
Okay, that's what we had thought.
Yeah.
I have also a quick question about this, which is just kind of with the. Actually, I'm going to reserve my question for the end. You can go right ahead.
Okay, sounds good. And to maybe complicate the ecological picture here, when we look at these two main clades, this top clade being Discinaceae and the bottom clade being Gyromitriae, we see an interesting pattern. Whereas all these Discinaceae clades, the spores, they do not germinate on standard media, and they have not been isolated from plants or lichens as endophytic or endolichenic fungi. So when we take these mushrooms, these fruiting bodies, and we take the spores and we try to grow them out in culture, none of these in this top clade grow out on media, on standard media. And that contrasts with this bottom clade shown here, where all of these will grow out in culture, including the truffles, Hypnotria, and they are readily found as endophytes or endolichenic fungi of plants, particularly mosses and lichens. So what this suggests is something interesting called the waiting room hypothesis, which is that oftentimes we see that mycorrhizal fungi will evolve from endophytic fungi. And that's because endophytes will kind of have the repository or the repertoire of genes that prepares them or makes them more likely. They already have that association with the plant, so they're more likely to then evolve a mycorrhizal relationship. So this is pretty interesting because we see a similar pattern here. And it might mean that even though we refer to these, or these things have a genomic signature of saprotrophy, that these other clades here in the tribe Gyromitriae, there might be a more complicated ecological picture here that we haven't quite fully understood yet, serving as endophytic fungi with mosses and other plants, as well as living within lichens. And a lot of this work comes from the lab of Matt Smith, Roseanne Healy, and Betsy Arnold, who will take mosses and lichens and they'll surface sterilize them, they'll put those on media, and they'll see what grows out of them. And there's a lot of fungi that seem to live within these lichens and mosses and other plants. So pretty cool and pretty strange. So in summary, we inferred a robust phylogenomic tree for the Discinaceae family, and we revised the taxonomy. We found that the mycotoxin gyromitrin has a limited and discontinuous distribution that is suggestive of horizontal gene transfer within the family, or perhaps from outside the family into the family, or maybe convergent evolution. And finally, Hypnotria appears to be the only mycorrhizal genus in the family, and all the others seem to be saprotrophic / maybe some sort of endophytic ecology. So what I'm going to talk about with you now is the taxonomic revisions that we made so we can learn more about these different genera within the Discinaceae family. And let's see, are there any questions in the chat? And yeah, thank you for clarifying what endophytes are. Great. Any other questions before I move on? Nope, that's all right. So what I'm showing you here again is Discinaceae, or sorry, family Discinaceae, and the two tribes, tribe Discinaceae and tribe Gyromitriae. So we're first going to talk about tribe Discinaceae, which includes 41 species. These are not formally described species, but these are all the species that exist within the sequence data. Many of them are undescribed, or we're not exactly sure what name corresponds to them. So I'll talk about that a little more. But first up, we're going to talk about the genus Discina. And as you remember, I said that a great thing about this system is that every single genus is identifiable by morphology. Typically a combination of macromorphology and micromorphology. So this group, just like many other Pezizales groups, typically does require microscopy to identify it with certainty to the level of genus or species. In the case of this first genus, Discina, these are these cup fungi, and they produce these really cool spores that have these points on them, and we call these apiculi. So they have these pointy apiculi. They have this kind of ornamentation, this network-like ornamentation, and they have these three droplets inside the spores that we call gutules. Okay, so a lot of vocab there, but apiculi, ornamentation, and gutules. And we'll see that the spores of different Discinaceae genera are modifications of these different features. And what I'm going to show you here are typically all the species that are known, and I'm going to highlight in brown the ones that are known to occur in Wisconsin or likely to occur in Wisconsin. So within Discina, this discoid genus, there are 12 species. You can see that a lot of them are only known with these temporary provisional names that are being utilized on iNaturalist. And only a few of them are actually known with their full species epithet. So within Wisconsin, we see Discina spa Michigan 01, as well as Discina warnii. And just a quick, I'll just explain this a little more. For people who are not an iNaturalist or people who haven't seen this in iNaturalist yet, particularly through the lab of Steve Russell, Mycota, and the MycoMap network, there is this system of provisional names that are being attached to specimens, to populations, to be able to refer to them because we know that this thing is a species, but we don't have a good name to apply to it yet. And that might be because it's an unknown or undescribed species, or because perhaps there are existing names, but we can't yet match those names to these sequence populations. So there is this growing system. If you're on iNaturalist, you're going to start to see these names more and more because they have been incorporated into the iNaturalist taxonomy, which is really exciting. So you might start to see names like this, and these are all provisional or temporary names attached to a species. So in Wisconsin, I expect that there's these two species you might be on the lookout for. However, this genus Discina is in sad shape. As you can see, there's a lot of unknown species. There's a lot of old names in this group. A lot of these species were described in the 1800s, and we have no idea what these names correspond to. And there's actually a whole, just like Pezizales, for example, Discina at some point was kind of a catch-all category for discoid fungi. So there's a lot of species with the name Discina that have no morphological information, no sequence data. We don't know if they belong in Discinaceae. We don't know what they are. So it's an old group and it's difficult to know exactly what's going on in this group. And I just want to show you here these two photos of Discina Michigan 01 as well as Discina warnii. This is a species warnii that was described from New York. And even though we don't have type sequence data, we're pretty confident what it corresponds to based on ecology and morphology. So I'd like to highlight these two specimens collected by Dave Wazalewski. These were collected on the same day at the same location. And even though the photos maybe have a little different coloration, they looked pretty much identical. However, when I sequenced them, we saw that they were less than 90% similar in the internal transcribe spacer barcode region. So these are way distantly related. And this just goes to show that there can be a lot of cryptic diversity hiding within fungi that look, to our eyes, very similar. So you have to really dig into the sequence data and the microscopy to start to see differences. Otherwise, they just both look like kind of wrinkly, large brown cup fungi. The next genus I'm sharing with you here is Moblancomyces. I know that's a bit of a mouthful. And these ones can be identified by their macromorphology as well as distinctively their spores have a blunt apiculus. So unlike Discina, which had a sharp pointy apiculus, these ones have a blunt one. And Moblancomyces includes discoid species as well as stipitate species. The discoid species, there's actually none that occur in Wisconsin, as far as we know currently. And within the stipitate species, there's about 12 that exist in the Moblancomyces corfii species complex. So you might know this as Gyromitra corfii, Gyromitra gigas maybe, or Montana, but Moblancomyces corfii is the current name and the main species that exists in Wisconsin. Although there are two other species that specimens like this that look like this could be in Wisconsin, which are Americana gigas, corfii, and Montanus. Montanus is typically a Western species, but it actually has been documented from the Midwest. So the discoid species definitely needs some revisionary taxonomic analysis, but the stipitate species, the Melblancomyces corfii species complex or the Gyromitra gigas species complex, has been extensively studied over the past few years, and we've made good sense of that group. So I'm showing you here pictures of these fungi. We have Moblancomyces corfii on the left. We have Moblancomyces montanus here. This is from Oregon. And then we have this Moblancomyces americana gigas. So these species, even though these three photos can look maybe different from each other, if you look across the variation within these species, you can't actually identify them morphologically. So these are species complexes that can only be separated out from ITS sequence data. So it's totally fine just to call these things Moblancomyces corfii because that seems to be the predominant species in the Midwest. These are all edible species. They have not been documented to contain gyromitrin, and there is a long culture and a lot of people who do eat them without getting sick. So all the evidence points to the fact that these are edible. With proper preparation, just like morels, if these are thoroughly cooked, people really do enjoy these mushrooms and they are widely foraged. So someone might ask, why the heck did you pick that name, Moblancomyces? What does that even mean? And I just need to say I did not come up with that name. So Moblancomyces was named after French mycologist André Maublanc, and the genus was created by Herder in 1950. So I just want to say, and I think this is kind of a misconception with this work that we did. People think that we created all these different genera. The reality is all these genera already existed because it's been a long and convoluted taxonomic and nomenclatorial history within this family. And a lot of these families, a lot of these genera, all of them except for one, already existed in the past. So a lot of us entered this group knowing a lot of things as Gyromitra. But if you go back a few decades, all these different names were actually in use. So what we did is, using the rules of nomenclature, we applied these names that already existed to the corresponding clades. So the only name that we actually created, genus name for this work, is Pezizales, which we'll talk about just in a little bit. Next group we have here is Neogyromitra, and these are really charming and fascinating mushrooms. Sorry, just looking at the chat real quick, and yeah, thank you for sharing those terms. Neogyromitra, these are really charming mushrooms, and microscopically, even though you don't need to look under the microscope to identify a Neogyromitra mushroom, they have these really cool spores with these kind of like little frilly or ciliate apiculi. So instead of having a single sharp or blunt apiculus at the end of the spore, they have a lot of these little protuberances, if that's how you say that word. And within this group, there was a study published not too long ago, last year, on the new Gyromitra group, still called Gyromitra at the time. This genus includes 3 discoid species, as well as 3 stipitate species. Neogyromitra brunea is the species that is most commonly going to be found within Wisconsin. There's also Neogyromitra caroliniana. It has a more southern distribution. However, I think there's two records of it in Wisconsin, and it might be moving over time with climate change. We might see it more at these higher latitudes. So even though Neogyromitra caroliniana, Wisconsin is pretty much out of its range, it does extend just a little bit into Wisconsin, and that might change in the future. Looking at these two species, these are exceptional edibles. Again, gyromitrin has not been documented in these species. They are widely consumed without any adverse poisoning events as long as they're thoroughly cooked, just like morels. And Gyromitra brunea, or sorry, Neogyromitra brunea, which I like to call the elephant ear lorchel, has these seams on top, and that's really iconic for that. Whereas Neogyromitra caroliniana, also called Big Red, can be a massive mushroom with these convoluted folds with this distinct reddish brown color. Again, both excellent edibles. Yeah, nubules is another great term for the protubeраtions. So then we get to this genus Pezizales. This is another group of cup fungi, and you can tell this apart readily with microscopy by looking at the spores. These ones have these really cool spores that have the kind of a scooped out apiculus. So instead of these projections at the tips of the spores being sharp or blunt and rounded, they're scooped out. And this is called a fishtail apiculus, which is how this genus got its name, Pezizales, which basically means fish cup, in reference to the fishtail-like projections on the spores. And they also have ornamented spores with these kind of like uh, wardiness to them. So this genus has just three species, and one of them I described back in 2024 from the Northwest, Pezidicina persicula. In Wisconsin, you might find Pezidicina leucoxantha. There's another species that's been described from the Northeast called Pezidicina convoluta, and this might just be a mature form of Pezidicina leucoxantha. I don't know if we're ever going to resolve this actual question. As you can see, this is the type packet from the fungarium, from the museum, that contains the specimens. And you can see this type packet specimen for what it was described as Discina convoluta. There is a note here that says destroyed. So there's no more specimen left. We're never going to know exactly what this specimen corresponded to. So the best bet is to go back to the type locale or the place where this mushroom was first collected to see if we can find something that morphologically looks similar and see if it's something distinct or if it's just actually Pezidicina leucoxantha, which is shown up here on the top left. Pezidicina leucoxantha is yellowish, and it's a quite rare species actually. So if you found this, it would definitely be something to note and hopefully collect and sequence. On the bottom left here is Pezizicina persicula, and this means little peach Pezizicina because it has a peachy color. And this was a species that Michael Buke talked about in his book Ascomycete Fungi of North America. He recognized it as a undescribed species, and it's something that we worked together to describe back in 2024. And finally, within tribe Discinaceae, we have Pseudodiscina, and this is another discoid fungus. This only has two species, only found in the Pacific Northwest and China. These have really interesting spores that are different from the others in the tribe. They have these two gutules or two oil droplets, and they have no apiculi. So again, just two species, but they are really cool. And if you're ever out in the Pacific Northwest, this is a pretty common species that you might find. Great. And then with that, we're going to move on to our next taxonomic group, which is tribe Gyromitriae. Scupiculus and Mortations. Oh my God, these are good words y'all are coining. So tribe Gyromitriae, this has 59 species that have been documented with sequence data. So let's go through those. First up, we have genus Gyromitra. This is maybe the most familiar, the most iconic group of fungi within this family. You don't need microscopy to identify them, but they have spores that range from being smooth with just two gutules, two droplets, to having a really broad, rounded apiculus on either end of the spore. Gyromitra has 8 species in it that are known from sequence data, and these are spread out across the world with kind of strange distributions that we don't have to get into right now. Within North America, there's 2 species, at least in the United States. There's Gyromitra venenata, as well as this undescribed species from the Pacific Northwest. Gyromitra is also a really old genus, and there's a lot of poorly described and documented names that exist within this genus that are really difficult to evaluate if they actually correspond to Gyromitra or what the heck they are. So these are what we call forgotten names that are shown on the right. Maybe they're species, maybe they correspond to some of these unknown species on the left, maybe they're just synonyms. This is an old group and it's really difficult to do the taxonomic work. So question, yeah, Gyromitra antarctica is a species that I don't think it's been found in Antarctica, but it was described from Patagonia, so the southern tip of South America. So trending towards Antarctica, I would say. But then we also have Tasmanica, which is known from Tasmania. And then we have some unknown species that have only been found from Turkey or Mexico. And this is a theme in this talk. The more sequencing we're doing, the more species there are. We are far, far, far from having a clear picture on the full biodiversity of Discinaceae. A lot of these species are quite rare. A lot of them look quite similar to each other. And a lot of areas of the world have not been sequenced or documented very well. So I'm showing you here these 2 different species, the Gyromitra splendida Pacific Northwest 01. And this one is really common in the Pacific Northwest. Here I'm showing you an albino version of it, which is really cool. Love to someday identify or figure out what makes these albino. Yeah, what's doing that? And then we have here Gyromitra venenata. So I'm showing you here a drawing of a mountain range to represent the Rocky Mountains. What we see is that this species over here is predominant in the Pacific Northwest, whereas Gyromitra venenata is predominant in eastern North America. But we do find that venenata exists in the Pacific Northwest, and there has been one sequence specimen of this species in the Northeast. So it's never quite cut and dry. These species have really strange distributions, and we need a lot more specimens to actually figure out where their full range is, what the full range is for these species. Right. So yeah, it is complicated. Right now, it seems like sequence data is what's required to figure out what species you're working with. But I hope with further documentation and really detailed studies on this genus, which definitely need to happen at some point, we'll be able to identify either ecological or biogeographical or morphological traits to these species that will allow us to identify them in the field. But currently, we don't have a full picture of that. Next up, we have the single genus of truffles that exists in the Discinaceae family, which are the Hypnotria truffles. These have, even though you don't really need spores or microscopy to identify them, they have really cool spores. Some of them are very rectangular or blocky. Other ones are very nodulose with these really broad warts. They have quite an actual diversity of spore morphology. Hypnotria is probably the most speciose genus within the family. We have documented 39 species with sequence data, and I guarantee you there are far, far more of this. Truffles are difficult to work with. Not a lot of people are finding truffles or collecting truffles. There are a lot of unknown species in Hypnotria, and there's a lot of singletons as well. What I mean by that, there's a lot of studies where you have a single sequence that came from an environmental data set. For example, they were sequencing the ectomycorrhizal fungi in a certain area, and you see that Hypnotria is extremely abundant and there's a lot of diversity. So there's a lot more work that needs to be done on this group. The only type specimens, the taxonomic, the nomenclatorial type specimens that have been sequenced belong to species that have been described from China in the past decade. So it's really impossible to know which names to apply to North American species. A lot of these are old European names. There's a lot of polyphyletic species concepts. Basically, it's just a mess, and there's a lot of unknown, undescribed species. So what I'm showing you here is that there are 0 Hypnotria specimens on iNaturalist from Wisconsin, which was quite surprising to me. They certainly exist in Wisconsin. There's one unknown species that is known from Minnesota. So it was first documented from Minnesota, giving it this code here. So we know there's going to be Hypnotria um, in this area, but none of them that have been collected and documented and sequenced from Wisconsin in this modern ecosystem. So if you want a challenge, I highly recommend going out and looking for truffles, which you can do most easily with a truffle dog, not easy to come by. You can actually rake around trees to collect truffles. It is a little more destructive, but if you put back the duff, I think it can be worthwhile, and you can find some really, really interesting and exciting species. So what I'm showing you here are various Hydnotria specimens from the Pacific Northwest, basically. And a lot of these have been found by Heather Dawson. So Heather Dawson single-handedly has found and collected and sequenced so many undescribed species. And there seems to be a huge diversity in the Pacific Northwest. That's probably not unique to the Pacific Northwest. It's just that there's a lot of collecting happening in California and the Pacific Northwest. I expect we would see similar diversity in other places as well. Heather Dawson has a truffle hunting dog that is fabulous at finding all these different truffles. So yeah, basically Hydnotria is a really, really underexplored genus, a really cool group of truffles, and there's probably so many different species around the world. So yes, there's a comment about using a hand rake to look for truffles. That's right. This talk isn't about that, but I'll just quickly say, if y'all want to do as a group, learn how to do it. I think that'd be really fabulous. What I will do is I move the duff from the surface. So if that's like conifer needles or leaves to expose the mineral soil, and and then I'll rake that. And then I'll make sure to put everything back so you can't even tell someone was raking there. And you can find a lot of species that way, and there's a lot of exciting finds to be had looking for truffles. Next up, we have Paragyromitra. And these are interesting because you actually find them in the fall instead of the spring. Pretty much most of the others are spring fungi. Paragyromitra has spores that are similar to Gyromitra, although they have typically more pronounced rounded apiculi at the end. But they can be identified by this saddle-shaped, um, the Ascomata have a saddle-shaped hymenium. So within the genus Paragyromitra, there are 8 species, and the one that you'll find within Wisconsin is Paragyromitra infila. And this is a species that has a global distribution. It's all over the place, and it is actually typically found in sequence and environmental data sets as an endophyte, which is pretty interesting. This is a species that there are not reliable edibility reports on it. It has been reported in the past as being toxic, but we have not detected any gyrometrin in this genus. It may be that not all specimens produce gyrometrin, or it might be producing a different toxin. So this is a questionable genus. Harmaya, was really a predominant taxonomist within this group, described some of these species, and he noted the toxicity of the species Paragyromitra ambigua. So these are reliable reports because he identified the toxic species that someone had consumed. He described the species, but we did not find gyrometrin in the species, so it's unclear what that toxin might be. So I'm showing you here these two main species that you find in North America, Paragyromitra infila, as well as ambigua. And what I'm noting here is that there's this really cool color reaction that was found for Paragyromitra infila from this collection from California. We apply KOH to all kinds of things. Maybe Damon was the first person to apply KOH to Paragyromitra infila, and he found this like really bright red color reaction. So even in a super common species like Paragyromitra infila, there can be new discoveries to be had. And these small features like chemical reactions using potassium hydroxide or ammonium or iron salts can actually reveal traits that can be useful for field identification. So I like to show this photo to encourage people to always bring your chemicals with you, test everything, because who would have thought you would get this bright red reaction with Gyromitra inflow? And if we were able to test more species, maybe this is actually a reliable field identification characteristic to separate out the different Gyromitra species. So coming to the end here, we have Pseudorhizina. These are some really amazing and rare species. We have Californica, Spherospora, and one unknown species from California. And in Wisconsin, Spherospora is the one known species. This is a very rare species. In a lot of places, it's considered endangered, and it's a really beautiful species that I've never found. And on Michael Kuo's page for this species, he talks about how this is one of his bucket list items to find this species. And lastly, we have Pseudoverpa. And this is a species that was only described in 2018 from a burn site in Cyprus. So there's just one species. It is Verpa-like, which is where the name comes from. It's quite similar to some presentations of Gyromitra splendida. I'm showing you here, these are some splendida species from the West Coast. And you can see they have a similar kind of Verpa-like attachment of the cap to the stem or the stipe. And so Pseudoverpa, I think, can be difficult to identify. There's this one collection here, again from Damon, that is just phenomenally blue and gray. So this is a very rare species that we need a lot more collections to understand its distribution and species diversity. So I hope that wasn't too burdensome to go through those different genera, but hopefully that was useful for you to understand the yeah, always bring your chemicals with you. Hopefully that was useful for people to understand the kind of scope of the different species that exist in this family. Maybe if you didn't pick up on it, a lot of these species are known from the Pacific Northwest. And I think that is not because the Pacific Northwest is higher richness in terms of Discinaceae fungi. I think it's because there's a much more sampling effort. We have things like the California Fungal Diversity Survey effort. We have a lot of people collecting and sequencing from the Pacific Northwest. I think if we had the same sequencing efforts in other places, we would see a similar diversity, let alone if we expand this across the rest of the world. So hopefully in 10, 15 years, because it might take that much time, we're going to see an increase in the biodiversity of Discinaceae. We'll have a better picture of all the different species diversity that exists in this family. So before we close it up, I know I'm running out of time. I just want to provide some tips and techniques for people who might be interested in going out and collecting Discinaceae fungi. So if you're looking for Pezizales fungi in general, it's really good to look around moss and persistently wet and bare soil. And alkaline sites are especially interesting. So here's a photo I took in New York where we have this nice kind of vernal pool. We have some bare soil along that. And I found this Paragalactinia species with these beautiful spores. So this is a great habitat to find a lot of Pezizales fungi. And in moss, you can find all kinds of cool things. This is a collection from California, Paradisina californica. This is a species we just described this year as part of that study on Paradisina, collected by 2 community scientists in California. The next tip I have for you is try to accompany your collections with macroscopic measurements and microscopy, if you're able to do that, of mature, freely liberated ascospores, and include the don't include apiculi. Yeah, so you want to show the apiculi in those photos, but you're not measuring them. So it's important. There's been a lot of confusion in the literature over the decades. Some people will measure the apiculi in their spores and some will not. So it's really important to clarify this is the dimension of the spore and then this is the size of the apiculi. And here, a quick video I have of showing a little cup fungus producing a bunch of spores. So you can get spores from cup fungi and from lorchels in general, but it can be tricky. Particularly Discinaceae fungi, they can mature very slowly, but I would recommend this publication here that talks about effective field collection of Pezizales to get ascospores. And finally, if you can sequence your collections, We are really in a sequence revolution right now, and my top recommendation would be to utilize the free sequencing services being supplied by Mycota Lab through the MycoMap website and service. And you can particularly go collect during continental mycoblitzes for free sequencing, or you can become a paying member. So this is a map from MycoMap. If you don't know the MycoMap network, check out their website. They're basically the ones putting all the sequence data on iNaturalist. And if you're in one of these states, Wisconsin is not one of them, but you're close to Michigan, any fungus you collect, you can send to MycoMap for free sequencing. So even if you're in, if you travel outside of Wisconsin, keep your keep this map in mind because if you go collect in Indiana, you can go send those specimens in for free ITS barcoding, which is extremely useful for identification. And I want to bring your attention to these two events, the Summer Continental Mycoblitz. This runs from July, early August, as well as in the fall. You can collect any mushroom during this time and send it in for free sequencing. If you want to be sequencing things outside this time period, you can become a paying member of MycoMap. They didn't tell me, they didn't pay me to advertise this. I am a paying member because it's a great service and I sequence my fungi at other times of the year. So they're now offering this membership service where you can pay monthly and then there's a certain amount of specimens you can submit for sequencing every year. So I think it's a great deal. And if you find that Gyromitra mushroom, you want to know what it is, you can become a member and you can get that sequenced very easily. You submit it to MycoMap with your iNaturalist accession number, and then in a few months, you'll get the data back into iNaturalist. So in conclusion, in this talk, we've talked about lorchel taxonomy, ecology, and Gyromitra distribution. I hope you've learned something about all those different facets. We've talked about the Discinaceae genera that can be separated by macromorphology and spore morphology. And we talked about, I showed you that there's at least 100 species in Discinaceae. Currently, there's exactly 100 species known from DNA sequence data, but there are many more taxonomic novelties expected with increased sampling. Many areas of the world have little to no sequenced Discinaceae specimens. Australia, for example, is really undersampled. Asia in general, Africa, basically North America is the best sample, but even then it's mostly just the Pacific Northwest. And I expect with the same intensive sampling happening in the Western United States, like in California, we'll see a much greater diversity of discoid, Discinaceae fungi, of the truffles, and even of these kind of really charismatic lorchels, these stipitate fungi. And that is everything. So thank you so much for your time. And yeah, I know I went a little over, but if there's any questions, I would love to, we could spend maybe a few minutes talking about that.
Yeah, thank you so much, Alden.
Great
Awesome.
job, Alden. That was awesome.
Yeah.
Thank you.
We do have a question in the chat, which is from Sarah, who's going on a trip to Alaska soon. And she's asking what Gyromitra species have been found in Alaska.
Alaska, there is a lot. There are some known undescribed species in Alaska. Moblancomyces, like the Gigas group. There's actually two undescribed species that have only been sequenced from Alaska, and no modern collections. These are only old collections from the Wells and Kempton collection, who are two prominent mycologists from the mid 1900s. They collected a lot in Alaska, and so there's two undescribed Moblancomyces species in Alaska. There's also Paragyromitra arctica, and there's no modern collections of that as well. So, and not to mention like discoid species, Alaska is a huge place and there's at least two undescribed species and one hasn't been documented in decades species and probably much more there. So you have a lot. If you can find something in Alaska, that would definitely be interesting.
I put a little note what I thought you said. You can correct me if I'm wrong, just for her note taking.
Yep. Mm-hmm.
And then the take home is, yeah, collections from there would be very valuable, and we could help you get them sequenced, or you could do the MycoMap thing, which sounds great.
Yeah. Mm-hmm.
Ben King is asking, I'm curious about the history of the term lorchel. I've heard some Helvella called this too. Were they formerly grouped together?
Yes, Gyromitra used to be Helvella before the genus was separated, and lorchel is a German word, I believe. I don't actually know the full like history or etymology. I really came across it with Nancy Smith Weber, who used that term, and from Larch. Yeah, so I think it's as I said, it does have historical use, and I'm trying to bring it back. And I think it's a nice way to talk about the stipitate Discinaceae fungi. Um, they're common names. There's not going to be universal agreement. I'm just kind of sharing with you how I like to call these things. So feel free to use that system if you like. If you want to call them false morels, I won't get mad. I know there's people who will get mad about that, but they're if we can understand each other, then that's all that matters.
Yeah, I think Lorchel is a really fun name. Like, I really feel like it has strong song potential for some reason. I don't know.
I think it goes really well with morel as well. Like, oh
It
yeah,
does.
I'm looking for morels and lorchels, and hopefully I find one or the other.
Yeah, it rhymes. Let me see if we had questions in the further back, or if anyone wanted to unmute themselves and ask, please just go for it.
I was have a question about, you said you're on a quest to consume a thousand and one different species of fungi. Why 1,001? I don't know the backstory behind that. Just
Yeah, thanks for that question. So that's a little maybe pun. It's literally a one-up on Charles McIlvaine, who was the mycologist from the 19th century who published 1,000 American Fungi. And his nickname was Old Iron Guts because he ate everything. He ate things that we know to be like deadly poisonous, and he just ate them. He was in the Civil War. He got shot, and he just ate all the deadly mushrooms. So it's a one up on Charles McIlvaine. I honestly don't think I'll ever get there, but it's good to have a goal.
Yeah, I love that. And I guess the follow-up question is, I've been like in my area, I pick a lot of the Gyromitra brunea. Like that's definitely the most common one in the Milwaukee area that I've seen at least of them. But if you had to pick any of those that you've consumed to be your favorite, is there any one in particular that you like more so than the others?
Yeah, I mean, Neogiant Mitchell Brunea is, I think, pretty fantastic. I think it actually tastes really good. So, uh, that's the one I've most eaten. Honestly, for all this talk I'm doing on Discinaceae, I haven't found a lot of these species because within one person's area, you might not actually encounter a lot of these different species, which is why this work is challenging because you have to think about it on a global scale, right? When you're doing this, you're getting specimens from all over the world. It's really hard to be one person and be in all the different places at once. So personally, I haven't found many of these species that I'm talking about. Um, but yeah, great question. I like Neogyra Brunei a lot. And I forgot to mention in that last photo, I showed an albino Gyromitra that was collected just yesterday by Jen Stevenson. Is Jen still on? Yeah. So Jen shared that with me, and I thought that was really cool because one, you can still find Discinaceae fungi in Wisconsin, maybe more in the north part. But two, these albino lorchels are really interesting, and we don't know what makes them albino, and they're quite rare. So that's a really cool find, and I'm hoping that can get sequenced so we can figure out if it's one of the Splendida related fungi or if it's Venenata. So we'll see.
And Jen was actually asking in the chat, Jen Stevenson. So if I find something odd, like an albino Gyromitra, should I collect several of them if they're present for sequencing?
I think it's the same as any other. I think what we think of as a population might be kind of arbitrary, but like if they're all kind of growing together, you can just collect a few and consider them as like one collection.
Yeah. Well, and of course, if something does end up being a novel species, somehow it is usually nice to have more than one in the
Yeah.
type collection. Or at least that's what I've been told.
For sure. It does happen that people will collect multiple things and they actually end up being different species. As I was showing with the Dacina, they were found maybe, if not on the same log, like really close to each other, and they were distinct species. And that happens too, like we go back and we sequence different collections from um, different types, different type collections. So there's the holotype, which is the main specimen that carries the name. And then paratypes. Paratypes are other mushrooms that were studied in giving a name to something. And I've gone back and sequenced the holotype and the paratypes, and they're different species, which means the person who was describing that species was looking at multiple species in creating a species concept. So it's challenging.
Yeah, I have a quick kind of, I don't know, phylogenetic nerd question for that or taxonomy question. When that happens, are you naming, are you discarding the paratype? Like if someone were to write it back up in a big paper, or are they like, yeah, I don't know what happens.
Discarding like the title of being a paratype.
Yeah,
I think you would still call it a paratype for that thing, but it's not that species anymore.
it's no longer valid somehow. Yeah, that's a
Yeah,
weird.
it's like basically like as name something can be the holotype of a name that we don't use anymore. You know, it's still the
Sure.
holotype of that original basionym. So I think it could still be the paratype for that species, but it's not that species. But that's a good question. I'm not entirely sure. Um, so maybe I'll take one last question, or if there's a second last burning one, I'll ask Jacina ancilis. So Benjamin King asked about Jacina ancilis. So Jacina ancilis is one of these original species names for the group. We don't know what Discina ancilis is. Um, we don't have sequence data that can be linked to that original species concept, and it's basically just completely unknown what that species is.
So that's one where a neotype would need to be named.
Yes. Yeah, I believe that there's not a is ancilis pursoon or freeze. I don't remember, but basically Discina ancilis, I don't think a type collection exists. We don't know what the species corresponds to, so it would have to be neotypified, which means there would have to be a new mushroom collected that's designated. But it's really hard because these species described in the 1800s, the descriptions are two sentences. They don't talk about, they say it's kind of a brownish gray fungus, you know? So,
Well, and sometimes the sites that they collected from too, it's like what was once a woodland is now a parking lot, and
exactly, yeah.
all around is built up. And it's like, how do you even say, oh, this is close enough to what they called it?
Yeah,
It's
yeah,
very
totally.
tricky. Oh yeah, so my dad, my dad in the chat is advocating for me getting to ask my question at the end. But if we want to put them off because you're done, Alden, that's fine too.
No, I'll take one more. Yeah, if you have your last question. I don't want to keep people too long and I know it's getting late. So yeah, let's take one more question and we can end it there.
Oh, okay. So I kind of have multiple, but I'm going to go with my most pressing one and we can chat later maybe at some point. But okay, so you were talking about that phylogenetic tree and the low support, and you were putting it down to horizontal gene transfer, and especially given that you're talking about sampling gaps. I was just wondering if there was any reason why you're like ruling out the sampling gaps, or is that complicated, like go read the paper sort of question?
Kind of there's no sampling gaps, I guess, in that work. You know, in terms of taxon sampling, we got all the groups that we know exist. So, you know, there are some species that are not represented, but uh, all the main species groups or complexes are represented in that sense. So
Okay.
certainly taxon sampling or missing like major lineages can be a problem. And I'm sure there's probably lineages that I guess I'm not entirely sure, but it may be that there are lineages in the
I'm
family
yeah.
that
I
we
mean,
have.
like, even less like, oh, you have examples of everybody who is described, but you were talking about like, oh, the Midwest is undersampled. There's all sorts of, and then if we were to go to other places of the world, like Africa, like probably much more undersampled for these
Yeah,
things, and maybe that could tell some of the more full story.
absolutely. You know, we got most like the long branches, like the main lineages. We believe we got those represented, but it may be that there's totally like novel lineages that are very distantly related to other things that would change our inference of the evolution. That
Yeah,
could happen. And certainly there's a lot of parts of the world, as I said, that are not well represented. And it's totally possible for there to be these like deep, deep time, deep evolutionary branches that we have not captured yet. That could change the topology, that's for sure. Yeah.
Well, and then within that same question, so I'm trying to count it as the same thing. I know that for truffles in particular, I've read about, or in tuber in specific, gene duplication events being what is behind, say, like the odor becoming intense, that they already were producing this and gene duplication events make it more intense, basically. And I was wondering, this is a pretty esoteric question, but like, what does that show up like in this phylogenetic data? Like, would it show up as a distinct thing, or is it like they're repeats, so you wouldn't be able to tell that change?
So this actual like the phylogenomic analyses I showed are using single copy genes. Um, there certainly are duplication events and whatnot. I actually didn't explore that level of like comparative genomics. So definitely an interesting biological question, but in terms of the phylogenomics, we just use single copy genes.
Okay.
And that's kind of a standard thing to simplify it because the technical term for paralogs, like paralogs are difficult to model. It can be done, but you typically exclude duplicated genes and just include single copy because it makes life a lot easier.
Okay, that makes sense. I'm working on getting good at phylogenetics, but am very far from it. So it's really interesting to hear about.
It's a journey, yeah, for sure. But yeah. Well, I appreciate all the questions. I really thank you all for having me tonight. It's been a lot of fun to thank you for letting me ramble on about this group of mushrooms, and I hope to, yeah, see you all around.
That's great. We were already talking about, oh, now the crust talk gets to start because
I know,
a
it's
lot
so
of us
funny.
love crusts and love your site for that as
Oh,
well.
thank
So
you.
yeah, if people don't know, Alden has I think it's like crustfungi.org. I'll get it.
Pressfungi.com.
.com.
And
Sorry. Yeah, you deserve. There we go. For
it is a website
crustfungi.com.
that anyone can actually make profiles on. So if you have a crust and you get it sequenced and you do microscopy, I know that's a tall stack of things, but yeah, keep that in mind. If you have a cool crust that you sequence and do microscopy on, you can make a profile for it on the website. So please.
All right. So our takeaway message is all the cool mycologists are on crustfungi.com and are also talking about awesome things like what did we call them? The wartations and the scrupuluses.
That's right.
Thank you
All
so
right.
much, Alden, and we hope to see you maybe at NAMA. Just
Yeah,
live.
we'll see. We'll see. I hope to see you all around.
All
Take care.
right. Thank you so much. Good night. Good night, all.
Good night.